Proper seed development depends on the inherent properties of the seeds themselves, as well as the environment under which seeds are developing. To continue our lessons in this module on seed morphology and anatomy, we will now look at these factors that determine the quality of seeds that are produced by flowering plants. Seed development is affected by both the genetics of the plant as well as the environment. The natural variation that we observe in size, shape, color, as well as in other features of a seed are determined largely by the genetic makeup of the plant. To illustrate this point, let's look at seeds of various plant species and see how one differs from another. This is a picture of seeds of epiphytic orchids. By epiphytic, we mean that the orchids are living on the surface of other plants, mostly trees. They do not take away nutrition from their host plants, so they are not considered parasitic. Seeds of some epiphytic plants are 85 micrometer long and weigh only 1/35 millionth of an ounce. They're essentially the size of dust particles. Compare that with the seed of coco de mer, which is considered the largest in the world. Coco de mer is a rare species of palm that is native to the Seychelles archipelago in the Indian Ocean. Seeds of coco de mer can reach 12 inches long and weigh up to 18 kilograms. That is approximately the weight of a small child. The difference in the size of the seeds of the epiphytic orchid and coco de mer is due to the genetic makeup of each plant and is not due to the effects of the environment. Within species, the genetic makeup of a plant may also cause variations in the different features of seeds. This slide illustrates intraspecific or within species variation in the seeds of corn, rice, and castor bean. In this panel, for example, we can see different shapes of corn seeds that include round, oblong, and even triangle. This picture in the middle shows slight variation in the size of the seeds, with the seeds on the left being slightly bigger than those in the right. And then in this last panel, we can see variations in the color of the seed coat of castor beans. You can see red, brown, white, and even speckled seeds. The variations that we see in the size, shape, and color of seeds of different accessions or cultivars of the same plant species are controlled by the genetic makeup of the plant. Aside from the genetics of the plant, the environment under which the parent plant is growing also affects how a seed develops. These environmental effects are often observed in terms of changes in weight, size, and number of seeds. The following slides will show how environmental factors such as soil fertility and moisture, temperature, light, and even the position of the seed within the inflorescence or the plant affects the way seed develops. Let's first look at the effects of soil fertility on seed development. Soil fertility refers to the amount of essential and minor nutrients in the soil that are available for plant uptake. Nutrients such as nitrogen, phosphorus, and potassium are considered essential nutrients because they cannot be replaced by any other element. In their absence, the plant will not be able to complete its life cycle. In contrast, iron, manganese, zinc, copper, boron, and molybdenum are considered minor elements. These elements, unlike nitrogen, phosphorus, and potassium, are required by the plant only in minute amounts. In general, plants supplied with optimum rates of nitrogen, phosphorus, and potassium will produce larger seeds compared to plants that have not been supplied with optimum rates of the macronutrients. Among the major elements, nitrogen has the greatest effects on seed development. Nitrogen is an important component of molecules such as DNA and amino acids, as well as chlorophyll. Chlorophyll in particular is important for plants to produce food through the process of photosynthesis. Depriving plants with nitrogen is equivalent to depriving plant with food necessary for seeds to develop. In general, the stress imposed by the lack of nutrients in the soil to the plants is referred to as nutrient deficiency. This slide illustrates the effects of nitrogen, an essential element, on the development of canola seeds. As I mentioned earlier, the effects of environmental factors on seed development often manifests in changes in weight, size, and number of seeds. In this particular example, we are looking at the effects of varying concentrations of nitrogen on the number of seeds produced by the plant. We have here three nitrogen treatments: 0, 45, and 90 kilograms per acre. From these graphs, you can see that increasing rates of nitrogen from 0 to 90 kilograms increase the number of canola pods per plant, seeds per pod, and also the overall yield. As an essential nutrient, nitrogen encourages the growth and biomass accumulation of plants. When applied during the vegetative stage, the benefits of nitrogen carry over to the reproductive stage. Aside from nutrients, the amount of water in the soil also affects the development of seeds. Soil moisture refers to water molecules that are held in the spaces between soil particles. Water that is present in the upper 10 centimeters of the soil is known as the surface soil moisture. In contrast, water that is present in the upper 200 centimeters of the soil is known as root zone soil moisture. This is the soil water that is available for plants. The lack of water in the soil imposes stress on plants in the form of drought. Conversely, the presence of excessive water also imposes stress in the form of waterlogging. Drought usually results from extended periods of water shortage due to abnormally low precipitation in a given region. In contrast, waterlogging often results from extended rainfall, especially in low-lying areas. When the soil gets saturated to the point of anaerobiosis, this is when waterlogging occurs. Let's look at an example of how soil moisture affects the development of beans. This data was derived from the results of studies that determined the effects of both drought and waterlogging on the weight of bush beans and climbing beans. Bean plants were subjected to either stress during emergence, vegetative, flowering, pod setting, and seed setting stages. The orange histogram indicates the 100 grain weight of bush beans under drought stress, while the blue histogram indicates the weight of climbing beans under waterlogged conditions. You can use the legend here to understand the labels of the histogram. So for example, we have DS-E here. This just means bush beans that have been subjected to drought during emergence stage. And then if we look at this first blue bar right here, we can see the label WL-E. This just means climbing beans subjected to waterlogging during emergence. The green bar indicates the average 100 grain weight of bush and climbing beans that were not subjected to any treatments. So what information regarding the effects of soil moisture on the seed weight of beans can we deduce from these histograms? Compared to the control, there is an overall decrease in the weight of beans subjected to either drought or waterlogging. This result indicates the susceptibility of beans to both waterlogging and drought stress. The negative effects of drought and waterlogging were higher when plants were subjected to either stress at the vegetative or the seed setting stage. Temperature is also an environmental factor affecting seed development. It is the measure of the degree or intensity of heat as expressed in degrees Celsius or degrees Fahrenheit. Extremes in temperature can impose either heat or cold stress. Plants have varying responses to temperature stress. In general, temperate crops will have a better adaptation to colder temperature, whereas tropical crops will have innate tolerance to higher temperatures. This slide illustrates the effects of temperature on the size of wheat seeds. Wheat is a traditionally temperate crop and therefore requires lower temperature compared to tropical crops like rice, for example. In this panel, you can see the gross morphology of seeds harvested from plants maintained at 18, 25, or 35 degrees Celsius during seed development and maturation. Based on these pictures alone, you can see that increasing temperature resulted in the reduction in the size of the seeds. The higher temperature may have caused a rapid desiccation of seeds during maturation, thereby giving smaller seeds. Light is another factor affecting the development of seeds. The various properties of light, including day length, light intensity, and light quality, will have different effects on how a seed develops. For example, Reduced light intensity due to partial shading results in smaller, lighter seeds. Crops like carrots, peas, soybeans, corn, and clover all produce smaller seeds when grown under shade. Light duration can also affect seed development. For example, short-day plants like peas that are subjected to long days and long-day plants like rye that are subjected to short days have been reported to produce smaller seeds. Light properties not only affect the development of seeds of cultivated crops, even the development of seeds of non-domesticated species are also affected by light properties. One study conducted by one of my graduate students looked at the effects of the environment on the development of silver nightshade seeds. Silver nightshade is considered a weed, and you can see them growing just about anywhere. In the study, silver leaf nightshade seeds were collected from 4 locations, which include the Texas Tech Quaker Farm, the Texas Tech Rangeland in Erskine, a rangeland in Littlefield, and a residential area in Lubbock. Morphologically, you can easily observe the differences in the size of the seeds collected from the different locations. You can see here that seeds from Quaker are relatively bigger compared to those collected from the other sites. Seeds from the TTU and Littlefield rangelands and urban site look like they are of the same size. But if you look at this histogram in the right, you can easily see that seeds from the urban site are relatively lighter. Why are these seeds lighter? Examination of the general ecologies of the mother plant from which the seeds were harvested from showed that the urban site constitutes a small patch of alleyway located under the shade of 2 adjacent pecan trees near a fence line covered with creeping vine. So you can imagine that silverleaf nightshade plants in this site need to compete with the pecan trees and the creeping vine not only for water and nutrients, but also for light. An indication that lack of light is a factor in seed development is the fact that the mother plants were significantly taller than silverleaf nightshade plants grown in open fields. Plants tend to elongate when placed under shade in search for light. This tells us that light was a significant factor in the smaller size of the seeds from this location. Lastly, we have the position of the seed within an inflorescence as a factor affecting seed development. Seed size is directly correlated to the amount of food reserves and energy content of the seed. This in turn are related to the amount of assimilates produced and transported by the plant. Within a plant system, organs are considered as either sources or sinks. The source refers to the organs that produces food. The leaves are perfect examples of sources. On the other hand, the sinks are the organs where the food needs to be translocated or transported. What are examples of sinks? We have the roots, we have the flowers, the fruits, and also the seeds. As the leaf photosynthesizes, it produces food in the form of photosynthetic assimilates. These photosynthetic assimilates are usually transported first to the closest sink or the sink that developed earlier. Let's look at the plant Pusidanum aureocelinum as an example. You can see in this picture that flowers of this plant are composed of several heads called the umbels. In this illustration, we can see 1 and 2 umbels. The bigger one is the primary umbel, and the smaller one is the secondary umbel. In this illustration here, we have the primary umbel on top, right here, and the ones that develop from branches here, 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 and here are considered the secondary umbels. Now each umbel is in turn made up of umbellets. The 2 pictures in the left show you umbellets within an umbel. In this picture, for example, we have 1, 2, 3, 4, maybe 16 to 18 umbellets. So when photosynthetic assimilates are transported to the floral structure, they will first go to the primary umbels. And within an umbel, the outer umbellets will be the first to receive photosynthetic products before the central umbellets, just because these organs were produced first. Now, the data in the right illustrate how photosynthetic assimilates are transported within a flower. In this first histogram, you can see that the primary umbel produced heavier seeds compared to the secondary umbel. And this is expected because normally photosynthetes are transported to the umbels that develop first, and those will be the primary umbels. The second histogram shows heavier seeds from the outer umbellets within an umbel. Again, this is because the outer umbellets would have developed first compared to the central umbellets. Let's look at another example of how position in the inflorescence affects seed development. This time we have rice. Rice is a crop that produces an average of 12 to 25 tillers depending on the cultivar. Each shoot that you see here is considered a tiller, and each tiller produces a panicle. Now let's take a closer look at the structure of the rice panicle. Within a panicle, we have a primary branch. This whole thing here is the primary branch. We also have a secondary branch, abbreviated here as SB, and a tertiary branch, abbreviated here as TB. During translocation, photosynthetic assimilates will first go to the primary spikelets in the primary branch before they are transported to the secondary and tertiary spikelets. Additionally, the panicles produced by the tillers that came out first will also receive more photosynthetic assimilates compared to panicles from late tillers. In summary, the development of seeds of flowering plants are conditioned by both the genetic makeup of the plant as well as the environment. While the genetics of the plant controls variations in several features of the seed, the effects of the environment are mostly manifested as changes in seed weight, number, and size. Some of the environmental factors affecting the development of seeds include soil nutrient and moisture, temperature, light, and even the position of a seed within the inflorescence or the plant itself.